Welcome again to AJP-Heart and Circulatory Physiology's video table of contents, or From the Editor's Desk. In the May 1st issue, we publish 18 original research papers, 1 rapid report, 4 editorial focus articles, and 1 editorial. I've chosen to focus this video on 4 papers that are the subject of the editorial focus articles. So let's get started. Zola et al. Determined if PPAR gamma stimulation improved skeletal muscle oxidation in mice with myocardial infarction-induced heart failure. Mice were randomized to receive a PPAR gamma agonist or placebo. After 8 weeks, skeletal muscle function was evaluated. PPAR gamma stimulation increased fatty acid oxidation and ATP levels in MI mice. Fatty acid oxidation was increased in the treated mice. PPAR gamma stimulation improved running endurance. In cell culture experiments, PPAR gamma stimulation was protective against cytokine-induced decreases in fatty acid oxidation and changes in metabolic gene expression. Pharmacological activation of PPAR gamma could provide a countermeasure to skeletal muscle dysfunction in heart failure. To examine the issue of renal afferent nerves in mediating the sympathoexcitation in heart failure, Zhu et al. Recorded action potentials in paraventricular nucleus neurons that project to the rostroventrolateral medulla, the site of presympathetic neurons in anesthetized rats. They found that 84% of these neurons were activated by renal afferent nerve stimulation. Neurons that were activated by afferent renal nerve stimulation were also responsive to baroreflex and cardiac afferent input. These neurons were glutamatergic in nature as they were activated by NMDA and inhibited by AP5. These experiments demonstrate that sensory information originating in the kidney is integrated at the level of the pre-autonomic neurons within the PVN and provides a physiological substrate for the effects of renal denervation in disease states. Wu et al. Hypothesized that cathepsin B plays a role in pressure overload cardiac remodeling. Cathepsin B was upregulated in response to hypertrophic stimuli in vivo and in vitro. Knockout of cathepsin B attenuated pressure overload-induced hypertrophy, fibrosis, apoptosis, and dysfunction. Aortic banding induced activation of the TNF-alpha ASK1 junk pathway was blunted by cathepsin B knockouts. These studies suggest that cathepsin B functions as a modulator of the hypertrophic process. Finally, Garg and colleagues tested the hypothesis that beta-adrenergic stimulation and rapid heart rhythm accelerate electrical failure in the globally ischemic heart. Optical mapping experiments were performed in isolated rabbit hearts subjected to 30 minutes of global ischemia. Hearts were paced in the presence or absence of isobutyrenol. They found that rapid heart rhythm and beta-adrenergic stimulation synergized to accelerate the decline in the optical action potential upstroke velocity and the progression of inexcitable regions. Both rapid heart rhythm and beta-adrenergic stimulation shortened the time to ventricular fibrillation during ischemia. The authors conclude that beta-adrenergic stimulation and rapid heart rhythm are major factors of electrical depression and failure in the globally ischemic heart and may contribute to adverse outcomes of sudden cardiac arrest. Thank you again for listening to this week's video table of contents. If you find this useful, please let us know and tell your friends and colleagues. We'll see you again in 2 weeks, and don't forget to send us your best work now.